All right, anatomy students, this is going to be video lesson 3 in the series covering the muscular system. And today we're going to talk about the synapse and how the neuromuscular junction and how the electrical signal from your brain is converted into a chemical signal that your muscle can use. And also we're going to talk about how some areas of your body have a very high number of nerve cells per muscle cell, and how in some areas there's a very low number of neurons per muscle cell. So that one neuron can control 2 or 3 muscle fibers, and in other places one neuron will have to control 2,000 or 3,000 muscle fibers. All right. And that's what we're going to talk about today. And it's a pretty long one. So buckle up and let's get her going. All right, everybody. Welcome to Muscular System video lesson number 3. Today, we're going to discuss a little bit about how the muscle contraction we talked about yesterday in the second video, how the body communicates to the muscle and tells it to start to contract. So we discussed how the muscle contracts. So how does the brain tell the muscle to contract? That's what we're going to look at partially today. So let's go ahead and get started. Excitation to contraction coupling. So we see a diagram here of the brain, and we see the pathway, the efferent and afferent pathways, effector and affector, how the brain sends a signal through the spinal cord. So we see the brain here sends the signal through the spinal cord. This is a section of spinal cord, and then out of the nerves down the arm, and tells your muscles how to move your pen across the paper to make the letters for the word you are writing down at this moment. How does the electrical signal from the brain get converted into chemical signal in the muscle? And that's what we're going to talk about today. Excitation contraction coupling, or EC coupling, involves events in the junction between a motor neuron and a skeletal muscle fiber. So we are looking at a picture through a scanning electron microscope that, as you can see, has been magnified. Scanning electron microscope is what that stands for down there. And it has been magnified 1,650 times. The microscopes that we have here are compound light microscopes. The highest magnification that you can get is the oil immersion lens, and that would be 1,000 times magnification. So this picture is 650 times more magnified than our most powerful microscope we have here at West Clermont High School. So we're dealing with small stuff here. So here's what we see. We see the nerve cells coming in, axon. So that's your nerve cell coming in here, and you can see it joins the muscle. This is a muscle cell, a muscle fiber. This is a muscle cell, a muscle fiber. And here again, you can see the axon branch coming down, and it joins the muscle right here. All right, so what goes on at this junction that enables your body to convert the electrical signal that your brain sends down the nervous tissue, all of this here, the signal comes down this way. And how does your body convert that electrical signal into a chemical signal that allows all of the stuff we talked about in lesson 2, allows all of that to happen? All right, that's what we're focusing on today. So this is a real picture through a scanning electron microscope of where the nervous system meets your muscular system. So if we magnify that and we clean it up with some wonderful artwork, if we magnify that, here's what we see. An enlarged view of the neuromuscular junction. The presynaptic, synaptic means where this joins. A synapse is a junction between two sets of either two neurons, where a neuron passes a signal to another neuron, or where the neuron passes a signal to a muscle. It's that space, that junction is called a synapse. And you can see here, this space between is called the synaptic cleft. You will also sometimes find it in different resources. You will see to it referred to as a synaptic gap. But this is what we're looking at, the space, the area between the nervous system and the muscular system, called a synapse. The presynaptic membrane, so this is in front of or before that gap, is on the neuron, while the postsynaptic, so if we're dealing with the direction in which this signal goes, so the synapse is the gap, the presynaptic, that's going to be the nerve, the postsynaptic on the other side of the direction of the signal is going to be the muscle. The postsynaptic membrane is the motor end plate. And again, this is where the muscle is going to be connected to the nervous tissue. And this plate is found on the muscle cell. There are two membranes separated by a space called a cleft or a gap. And we typically refer to that as the synaptic cleft or synaptic gap. Conscious thought. This is you, your consciousness telling you to raise your hand or to grab your phone and look at the screen because you got a notification. This is all up to you. Results in activation of a motor neuron and release of a neurotransmitter. Neurotransmitter that we are talking about, that our bodies use, is called acetylcholine. That is this word right here. It is typically abbreviated as ACCH. Sometimes you'll see it as just ACH without the little lowercase c. Kind of depends on, again, the book you're looking at. So acetylcholine is the neurotransmitter, and it's going to be used at the neuromuscular junction. NM is neuromuscular junction. So what happens is all of this acetylcholine is stored in the nerve cell. You can see the nerve cell right here, and all of this is acetylcholine. When the electrical signal, here you can tell because you got the little lightning bolt there, that's electricity coming. The electrical signal reaches this end of the neuron. This neurotransmitter is released. This chemical is released, and it travels across this gap, synaptic gap, and is picked up. So it goes from synaptic vesicles to synaptic receptacles. It travels across and is picked up by those synaptic receptacles. And that will then tell the muscle, hey, we picked up these little acetylcholines, and that's going to tell us to do our job and contract. All right. And to tell your muscle to stop contracting, we have an enzyme called acetylcholinesterase. Remember, anytime anything ends in ase, it's going to be an enzyme. So acetylcholinesterase comes in and will remove acetylcholine from those receptacles and inactivate it to tell your muscle to stop contracting. So it just isn't used up and thrown away. There's an enzyme that has to come in and remove the acetylcholine to tell your muscle to stop contracting. The plasma membrane on the far side, or the muscular side, belongs to the muscle cell and is called the motor end plate. The motor end plate is rich in chemical gated sodium channels. Remember from biology class, there are proteins embedded within your cell membranes. Some of them are marker proteins. Some of them are gated channels. And a gated channel, of course, is a protein that is used in what is called facilitated diffusion. So when they are, normally they're closed, but when they are given the signal, they will open, and they're floodgates that allow substances to now rapidly diffuse across the cell membrane. So instead of having to find their way and mitigate their way through the phospholipid bilayer, these are actual doors or gates that will open and will allow these materials to diffuse quickly from the outside of the cell to the inside of the cell. So the motor end plate is rich in chemical-gated sodium channels that respond to acetylcholine. Another way to say this, the receptors for acetylcholine are on the ligand-gated sodium channels on the motor end plate. All right, so here's what that means. I know that's very wordy. So think of acetylcholine as a key, and think of the acetylcholine receptacle on the muscle has the keyhole to open a gate. So acetylcholine comes in, fits, this is the key, fits in the keyhole here and here, and when it fits in the keyhole, it unlocks the gate, which opens and allows substances then to rapidly diffuse across the cell membrane. Normally, because these ions are these calcium ions and sodium ions are too big to easily get through this phospholipid bilayer, they need a place to go, and they need to get there quickly. So that's what the gated channel is for. When they receive the key, the acetylcholine, it unlocks the gate, and the gate opens, and the sodium and the calcium will rush in, the sodium will rush in, and the potassium will rush out. And that's how the gated channel works. The chemical events at the neuromuscular junction transmit the electrical events of a neuronal action potential into the electrical events of a muscle action potential. What that means is the acetylcholine traveling across, that's the gap that allows your body to, that's the go-between that will allow your body to convert an electrical signal that says, the electrical signal says, hey, muscle, contract. And once that electrical signal gets to the end, the acetylcholine will travel across the junction between nerve cell and muscle cell, and will then, that's the converter that converts that electrical signal that says muscle contract into a chemical signal in the muscle, almost translates that signal into a chemical signal in the muscle that says, hey, muscle, contract. Once this happens, we get what's called a muscle action potential. That's what the AP is short for. It is propagated over the surface of the muscle cell membrane through what are called voltage-gated sodium and potassium channels. So here we have another protein that is a gate that will allow for the quick movement or diffusion of sodium and potassium that will allow your muscles to contract. And once the muscle reaches the action potential, so we're going to have a graph that shows what that is, then these gates open, and the sodium and potassium gates will open and will allow the potassium to flow through that gate, and that changes the muscle cell and allows it to do its job. By placing a micropipette inside a muscle cell and then measuring the electrical potential across the cell membrane, the phases of an action potential can be graphed in this figure. So here's how this works. Here's what we're looking at. Acetylcholine comes across, and it will tell the cell, hey, we need to contract. The muscle cell, though, won't contract until enough of those ions flow across the membrane to change the electrical charge of that muscle cell, or depolarize it until it reaches a millivolt of plus 30. If the acetylcholine rushes across and the ions dump in, and there's not enough to change it to plus 30, then the muscle contraction doesn't take place. It only takes place when it reaches the action potential. If there's only enough ions to change the polarity to 0 millivolts, from negative 70 to 0, that's not enough to trigger an action potential. That's not enough to cause the muscle to contract, and it won't contract. But once it hits the action potential, once it hits a charge of plus 30 millivolts, then that's what causes the muscle to contract. And then it will have to pump out all of those ions to bring the charge back down to negative 70 millivolts. And it will then repolarize and reset again so that it can contract again. And all of this stuff, here's the part that just boggles my mind. All of this stuff that we've been talking about that happens, biochemically inside your body in the last 2 video lectures, all of this stuff happens in as quickly as you can. Think about how fast you can contract a muscle and stop and then contract that muscle again. How many, if I say clap twice as fast as you can, boom, boom. All of that stuff that we've been talking about in these lessons, all of that happens that fast. And I just find that amazing. That's why I like talking about muscles. Skeletal muscle metabolism in a state of homeostasis, which is when everything is in balance in your body. Muscle use of oxygen and nutrients is balanced by the production of manageable levels of waste products. And those waste products in your muscles are primarily carbon dioxide, lactic acid, and heat. You're going to produce, I know it's weird to talk about heat as a waste product, but it's more of a, I guess you could call it a byproduct of your muscles contracting. This is how we keep ourselves warm. We maintain a constant body temperature. And one of the things we do to maintain this constant body temperature is we move our muscle cells. That generates heat, and that is basically the furnace that keeps your body warm. Think about it. When you get cold, you start to shiver. And the reason you're shivering is your body is taking over and saying, we need to generate heat. You're getting too cold. Let's do something about it. Let's kick the thermos up. Let's kick the thermostat up here, and let's get the furnace going. And so you start to shiver. Rapid muscle contractions generate heat to try to bring your body temperature back up. 70 to 80% of all of the energy used by muscles is lost as heat. This is why we have to eat. One of the if we talk about it, you've been in zoology and we talk about the metabolism of ectotherms, or commonly referred to as cold-blooded animals. How can a snake eat once a month and be totally fine? Well, they don't maintain a constant body temperature. It takes a lot of energy to maintain a constant body temperature. When you're a reptile and your body temperature fluctuates with the environment, you don't need as much energy, so you don't need to eat as much. Now, the drawback is when your metabolism is that slow and the temperature drops, now you become slow and lethargic. So we, as homeothermic or quote unquote warm-blooded animals, when it's cold, we are not lethargic. We can still move quickly and respond. Our reflexes are still there. Whereas a snake, when it's cold, it becomes very slow and sluggish and lethargic, and that can make it susceptible to predation. So that is one of the drawbacks of being ectothermic. Oxygen debt. When you exercise, your muscles are requiring or using oxygen faster than you can get it into your body. So you build up a debt. And this is why you still continue to breathe heavily after exercise, because you're paying back that debt. It is referred to as EPOC, or excess post-exercise oxygen consumption. EPOC is much easier to say, or just oxygen debt. This is the amount of oxygen repayment required after exercise in skeletal muscles. So if you're in shape, you won't reach oxygen debt as quickly as someone, like myself, who was out of shape. I reach oxygen. I start to have to repay oxygen very quickly. If you're in shape, you won't reach that point because your muscles are more efficient at using the resources and oxygen available. And if you do reach an oxygen debt, your body can pay it back quicker. If you're like me, it takes longer to recover. I have to breathe heavily. Let's say I climb a few flight of stairs or whatever, and it will take me longer to recover because I'm fat and out of shape. And what we do, the reason we have to repay this oxygen debt is to replenish ATP stores, replenish creatine phosphate and myoglobin stores, and convert lactic acid back into pyruvate so it can be used in the Krebs cycle to replenish ATP. And I'm sure that we all remember from biology class how pyruvate is used in the Krebs cycle system to replenish ATP. So there's no need for me to go into that. This brings us to the all or none principle. The all or none principle states that when an individual muscle fiber is stimulated to depolarize and an action potential is propagated along its sarcolemma, it must contract to its full force. It cannot partially contract. Also, when a single motor unit is recruited to contract, all of the muscle fibers in that unit must all contract at the same time. So what this means is when a neuron tells all of the muscle cells that it controls, when it says contract, all of the muscle fibers or muscle cells associated with that neuron, when they're told to contract, they will contract as hard as they can to their fullest extent, or they will not contract at all. There is no partial contraction. It will go, it's either nothing or 100% maximum power. There is no go between. So this brings up the question then, when I go to pick up a styrofoam cup, why don't I crush it if my muscle cells are going as hard as they can? And the reason is your body, using input from your touch receptors and pressure sensors, and using visual stimulation and context cues, your body automatically decides for you how many neurons it will use to tell how many muscle cells is needed to do a particular job. So when you go to pick up that open water bottle, everyone has the ability to crush that bottle and squirt water out of the top, but you don't, because your brain automatically recruits just the appropriate number of muscle cells it needs to pick up that bottle. Now, if you go to pick up a brick, you're going to have to squeeze tighter to pick that up, so your brain will automatically recruit a higher number of muscle cells, because your neurons are connected to thousands and thousands to do that job. You can program a robotic arm to pick up a cinder block, but if you put an egg in its place, it's probably going to crush it, because it doesn't have that kind of feedback mechanism to say, hey, don't squeeze it that hard. And sometimes it works against us. Have you ever go to pick up a cardboard box that you thought was very heavy, and it turns out it was just full of styrofoam peanuts? And, your brain says, ooh, you're using context clues and whatnot, this is going to be heavy. then you go to pick it up and it's not heavy, and you end up almost throwing it over your head. All right, so sometimes it works against us, but most of the time it's an amazing thing that our body is able to do. Motor unit is composed of a motor neuron plus all of the muscle cells it innervates, or how many it controls. So in different parts of your body, you will have one neuron, one nerve cell will control a high number of muscle fibers, or in areas where you need high precision, then you will have one nerve cell will only control a few hundred or a much smaller number. And those are in areas where we don't need power and we don't need bulk movement, but we need high precision. We need very finite muscle contractions. And examples of that can be controlling the tension of your vocal folds, for example. When you want to talk in a higher or lower pitch, or when you're singing and you want to change the notes that you're singing, that takes high precision. Moving your eyeball is a very high precision. Writing letters is high precision. Your quadriceps and your glutes, not so much high precision. So your neurons in your controlling your butt muscle, your gluteus maximus, they're probably going to have thousands and thousands of muscle cells for every neuron that they control. But controlling the movement of your eyeball, you're gonna greatly reduce the number of muscle cells each neuron. So you're gonna have a lot more neurons in any given muscle in areas of high precision than in areas of low precision. So high precision, few muscle fibers per neuron, such as the laryngeal and extraocular muscles, which is what I just said. Laryngeal, larynx, your voice box, and extraocular. Those are the muscles that move your eyeball. And low precision, many fibers in the neuron, your thigh or your glutes. You're going to have up to 2,000 to 3,000 muscle fibers for every neuron. So as an example here, these small muscles that move your eyeball, these are going to be extreme precision. So they're going to have very few, 1 to 4 muscle fibers for every neuron that's moving your eyeball. And if you've ever watched someone's eye when they read, which when we get to special senses, if we're back in school, we've got a little lab activity where you will do that. And you'll watch their eye will bounce as they read across a page, as their eyes take in chunks of words, usually 4 or 5 words at a time before the eyeball needs to move. And when you watch someone read, their eyeball just goes bop, 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 bop across the page. It's not a smooth wave that doesn't just pan smoothly across the page. It jumps. And the amount it moves is ever so slight. And that's what we talk about with high precision. All right, I think that's going to be enough for video lesson 3. All right, that's it. I told you it was a big one. Hopefully everyone is still conscious, and I didn't put you to sleep, and you were able to understand all of that anatomy jargon I was throwing at you. So if we are remote at this point, now that you're finished with watching this video and taking your very detailed notes, you now will go to Schoology and open the exit slip for video lesson 3 on the muscular system. And as always, because again, I'm a YouTuber, so I have to say it, don't forget to click like and subscribe. And if you have any questions, just shoot me an email and I'll try to answer them to the best of my ability. And this was a pretty confusing one, so I won't be surprised if there will be questions coming. So thanks for watching, and I'll talk at you guys later.